So it's a real opportunity for UTEP students to actually do cutting-edge research. These pathways of the brain have never been mapped to an atlas like this in the history of the field, which is pretty cool. I wish I were a freshman and taking my own course.
When do I sign up?
Yeah, my name is Arshad Khan. I'm an assistant professor in biological sciences and neuroscientist by training. Yeah, in our course and in my research, they're intertwined. Basically, the RDC is going to be, the students will be carrying out my research. And the research is really focused on the brain and brain circuitry. And we're trying to basically map new circuits and pathways in the brain. And the goal of the project is to develop the highest resolution maps of an area of the brain that really hasn't been mapped that much. It's in the base of the brain. It's called the hypothalamus. And so for humans, it's actually a very vital part of the brain. And there's a lot of disorders associated neurologically with a dysfunction of this portion of the brain.
All right, so can you tell us your name and what you do?
My name is Charles, Dr. How do people normally do it? Dr. Charles? My name is Dr. Charles Spencer. I'm an assistant professor in the Department of Biological Science. My research interests are in zoonotic diseases, so those diseases that you can get from an animal, and in bioterrorism agents. So we're trying to prevent the use of infectious disease as a bioterrorist weapon. But the research in my lab is focused on a particular bacterium called Francisella tularensis. It causes a human disease known as tularemia. As a bioterrorist agent, it can be released by aerosol, so all they have to do is fly over the city and spray it, and you can become infected with it. So we found that a particular cell type, called a natural killer T cell, may be responsible for controlling that infection. So the work that students that participate in my lab course will be directed towards identifying the molecule from the bacterium that these natural killer T cells recognize, so that we can use that as a potential therapy or drug target.
Okay, my name is Tuan Chou, but most students call me River because that's River is translation of my first name in Chinese. So many students even call me Dr. River. So we use biochemistry tool to study structural biomolecules, like those examples here. So the other direction of my lab now is to really focus on the circadian. So and every of our cell has also intrinsic biological clock. So and every 24 hours they change your cells, and all this clock was synchronized by our brain, a small area. And these clocks are very important. 60% of our prescriptive drugs are circadian related. That means you'd better take that drug at a particular time, has the best benefit for you. So these things are running daily in every of your cells. So, but the understanding how these molecules interact with each other, that's another big research direction now in my lab.
Oh my goodness, is that seismogram? Is that your son's?
That's my son's drawing of a seismogram.
Oh, that's so cute.
I don't know, about 3. So my name is Diane Douglas. I'm a professor in the Department of Geological Sciences, and my specialty is geophysics. So I'll be teaching a course that involves looking at the soils around El Paso, and we'll be making measurements of their geophysical properties as well as their geological properties. So we will go out in the field and measure like how fast sound travels through the soil. We'll measure how it conducts electricity, its magnetic properties, and we'll use this information along with information on the minerals in the soil to try and tell us about how salty the soil might get when farmers water it during irrigation. Also, how does water pass through the soil? So if we have an oil spill somewhere, could we predict how fast it might contaminate, say, a water supply in one area of town versus another?
Hi, I'm Novia Gran. I'm an assistant professor here at UTB in the Department of Chemistry. So in my lab, we're interested in multi-electron redox chemistry. And multi-electron redox chemistry are processes in which one or two electrons are transferred from one place to another. So the research that we're trying to do is we're trying to synthesize materials that allow us to make those type of reactions. So one of the most important problems that we have currently is the energy problem. We need to find ways, alternative sources of energy to the hydrocarbon fuels that the world is used to. So what we need to do is we need to understand the basic chemistry of energy conversion, and our work attempts to solve those problems. So as you can see in the lab, we are mainly a synthetic lab, so you will be learning synthetic techniques. And there are several techniques that people can utilize. Many of those that we do in here are air sensitive. In other words, we try to remove, to get oxygen and water out of our reaction vessels, and for that matter we have instruments like this.
But cobras can be a bit more aggressive than this kind of llama. So bites by cobras are not that uncommon. If you get bit by a cobra or a mamba in Africa, you're probably going to die.
What I basically do is I use DNA sequence data to understand the relationship of different populations of animals to each other. And the animals that I work on are amphibians and reptiles from Central Africa. My research program has been in the Congo for the last 9 years. So students will have an opportunity to work on the samples that I brought back from that field work to figure out how populations are related to each other, and species boundaries is very important with that research as well. So where does one species end and another begin? Because often if you're just looking at them, you can't figure that out. You need the benefit of the DNA sequence data, and that's what students will be working on for the course.
And that's a neuron. That's my neuron. My one neuron. I always tell students I have one neuron.
Just relax. Just relax.
All right.
Relax.
Uh-huh.
There we go.
Right. Hello. My name is Herman Acosta. I'm a virologist. I also teach the Phage Hunters Lab, which is aimed at freshman students. So it's a really different type of class. It's aimed at offering freshman students the opportunity to do real research. So the build class that I direct is the Phage Hunters Laboratory. And what students actually do in that class is they try to isolate bacteriophages, that is viruses that are able to infect bacteria, and they isolate them from our local environment. So basically what we are doing is looking for viruses that are present in our El Paso del Norte region. And what we try to do is look at how those viruses that are specific of our geographical area are different from viruses that are located in other places around the world.
This is actually the cutest one that I have. I have a couple of little West Niles that have little Egyptian eyes that are very cool. But you have to be really careful when you say, oh, I have a pathogen, or my friend gave me this disease, when you mean, my friend gave me this cute little stuffed virus. Okay, I'm Dr. Kyle Johnson. I'm an associate professor in biological sciences here at UTEP, and my lab studies how viruses reproduce their genetic material. So a virus, much like this one, are composed of a protein shell, and then inside that protein shell is nucleic acid. It can be DNA or RNA. And this is where the genome is in a virus. And so there are all sorts of complex mechanisms by which the virus has to interact with a host cell, inject its nucleic acid into that host cell, and then proceed to take the cell over. And that really is the basis of human disease, but also gives us the opportunity to study the basic science by which some of these processes occur. And so my lab studies the mechanism by which that genome replication occurs. And a lot of it involves cell biology and looking at how cells and viruses interact. We're really doing it within the context of forensic science.
Hi, my name is Manuel Yano. I am a professor at UTEP. I am a professor of microbiology, and then also I have a laboratory that do research in HIV. The research that I do is mainly trying to identify cellular proteins that are implicated in HIV replication. HIV is a parasite, is a microorganism that needs the cell for replication. So during evolution, HIV has learned how to use proteins in the cells in order to replicate more efficiently. So then the idea that we have is that if we identify these proteins that are cellular proteins, we will have identified new targets in order to inhibit viral replication. So then in this course, what we're going to be doing is generating mutations in the cellular proteins that we have identified in order to map what regions are implicated in HIV replication. So the students that are going to be enrolled in the course going to learn how to do mutations in proteins.
My name is Mark Cox. I'm an associate professor in biology. I'm a prostate cancer researcher, and I'm just an all-around awesome dude. How was that?
That works.
That'll work. In my own research lab, I study prostate cancer, but more importantly, we're trying to understand the mechanisms by which prostate cancer progresses to a more aggressive form, and trying to identify novel treatment strategies for treating late-stage prostate cancer. And in my laboratory, we're trying to target some of those novel mechanisms with small molecules or novel drugs for treating prostate cancer. So the course is largely designed around that research strategy, and that we've developed a number of novel drugs for treating prostate cancer. So by the end of the course, the students will have not only developed assays for screening drug libraries, but they will come up with their own novel drug molecule. It's a 2-semester course, and this assay that the students have developed to screen small molecule libraries, they will then extend the use of that assay in semester 2 for use in screening environmental samples.
Hi, I'm Dr. Charlotte Vines. I'm teaching 3 courses this year. One of them is developmental biology. The second one is G protein coupled receptor biology, and then the third one is mammalian physiology. We're interested in trying to control a number of different things that happen with the immune system. The receptor that we study, CC chemokine receptor 7, is involved in controlling all those processes. What we're interested in having our build students do is develop tools that can control CCR7. One of the tools is a protein antagonist, and that is what you would be working with in our lab and developing during your time in the build program. Children who get T-cell acute lymphocytic leukemias, a lot of times the tumors will migrate into the brain. And there's only one way they can get in, or one way that we know of right now, and that's with the protein that we study in our lab. So if the leukemic cells express this protein, they can move into the brain. Now the reason why you care is because if the cells get into the central nervous system, they can hide. And they hide from chemotherapy that normally would be able to kill them. And then when the children come off the chemotherapy, the cells move back out into the periphery, into their bloodstream, and they kill the children. If we can successfully develop this antagonist that I'm trying to get you guys to help us develop, and we can basically save their lives after they go through normal chemotherapy treatments.